I'm Jonah Comstock, Editor-in-Chief at Pharma Forum. I'm here at Reuters Pharma USA with John Androsavich, General Manager of DataPoints at Gingko Bioworks. John, thanks for joining me.
Yeah, absolutely. Glad to be here.
So give me the quick version of what the DataPoints unit does at Gingko.
Absolutely. DataPoints, I think, is the distillation of everything Gingko has built for the last 10 plus years. And so it's really taking our automated foundry, all the robotics that we have there, all of the data infrastructure that we have to pull data from those robotics, and providing that as a service to the industry so that the industry can build large multimodal data sets to train AI. This can be in discovery, such as building foundational models for understanding how a cell responds to different perturbations. It could be also for protein design, how to think about building the right antibody that has not only just binding affinity, but also the drug-like properties required to actually make it into a product. And so this is almost more CMC or early stage development, but doing this not for just a couple of lead compounds, but actually doing it for tens of thousands of compounds to allow you to build a training model that then can become much more predictive and allow you to make better decisions, not only for the campaign you're currently running, but for future campaigns.
So this is AI in the old sense, right? We're not talking about generative AI. This is large, big data sets, algorithms, models. How long have you been in this space? I mean, DataPoints, but also you.
Yeah. So DataPoints actually launched in September.
Okay.
So this is a new offering. I would say that it is tackling the bottleneck for bio AI where it really exists, and that is it's not in the GPUs, it's not necessarily in the algorithms, the architectures, it's really in the data. And so what we have is, for biology, the one thing that everybody points to, also around the time that DataPoints formed, maybe in October, is you had the Nobel laureate for AlphaFold, right? And so this was using the AlphaFold model that DeepMind had discovered, and along with Google, and allowing you to predict the protein structure based off the protein sequence. The way that actually came to be was because we have decades of data to go off of, highly structured data that protein crystallographers, structural biologists have actually been contributing to Protein Data Bank for year over year over year, and all in the same format. And so fortunately, we were able to mine that and use that for training AI to now make AlphaFold. The challenge for all the other domains of biology, and there's so many other domains, is that you just don't have that data that exists that is all structured and also labeled. And those are the really key fundamental components. And so pharma especially has such a rich history of data generation, and they have data all throughout their various departments. And some of it's siloed, so some of it's actually like a bureaucratic issue, if you will. But oftentimes it actually comes to the structure of the data. The data just wasn't meant to be used for AI training, and there just isn't enough of it that is kind of consistent that you can put together in aggregate to sum up to that meaningful training data for a model. And so sometimes the approach is just start from scratch. And with automation, the ability to do high throughput biology, you can actually ramp up your data generation intentionally in a very short period of time. And so that's really where it comes down to. It's actually data for the new age of AI biology, but it's really allowing us to generate new algorithms with the right type of data behind it. I've been in the AI space not too long. I think that we're all kind of adjusting to this new world. I have a deeper history in bioinformatics. I came from Pfizer before Gingko, where I was in business development, search and evaluation in RNA medicine space. And in RNA medicines, bioinformatics plays a really big role. There's billions, if not infinite number of different combinations that you can use to build an mRNA structure, right? And so this is kind of like the thesis here of AI and nucleic acids, is that you can use AI to actually pick the right sequence for mRNA. And we do some mRNA stuff in Gingko as well, including models that can actually spit out generative models that can predict the more stable structures for mRNA. So that's an example. And so before Pfizer, I was in bioinformatics at various biotech companies, running large screening campaigns, doing that therapeutic drug development. So I would say I've always been interested in large data sets, kind of that scaled approach to biology, understanding that if you take a couple of shots on goal, it just probably isn't enough to explore all the empirical space biology, but you really need to take large expansive campaigns in order to at least start to understand what could be the best combination when you're thinking about engineering the right parts and putting all these pieces together.
So then how do you take that idea of building sort of the AlphaFolds for all these other parts of biology that don't have clear data sets? How do you take that idea and make it into a business model? Who are your customers, and what kind of guides what you're working on and when and how you're prioritizing it?
Great questions. What we're really focused on is building up workflows that people already buy into. That is that we're not trying to prove that, hey, you should be doing this assay, or maybe this is a really good readout that you should consider. No, it's more about things that people are already doing, companies are already doing and spending money on, either internally or externally, but now scaling that up to a new level. Where it used to be, okay, where you're used to these 12 assays that we use, these biophysical characterization for antibody developability. These are the gold standard, and if you run these assays and it all comes at the end, you know you have a molecule that shouldn't cause you any problems when you get to the clinic. Now, there's always surprises, right? But in general, it's all agreed that these 8 to 12 assays are meaningful, right? And so the question is, how do we scale that up? How do you do this in the orders of tens to hundreds of thousands so that you can actually build predictive models off of it? And that's really where we're gearing all of our infrastructure that we have and now and mindset too and operations and putting that now towards building these data sets. And so we're not convincing people of doing something new per se, but we're convincing them to do it at scale now so that they can feed it into AI models. Similarly, we're working in spaces where with functional genomics, we're looking at genetic knockdowns, chemical perturbations, and doing this also in the orders of 384 well plates, running these through multiple campaigns, 96 plates a week. That's generating massive amounts of RNA sequencing data. So we can do RNA sequencing on a single well that is for $10. Like that is an incredible amount of data. 10,000 genes you're measuring for $10. So that's an incredible amount of data that you can get. So we're also thinking about what assays exist today, like RNA-seq, right? Pretty expensive. Transcriptomics. How do you do transcriptomics at scale? Well, we actually have an assay now that actually is much more valuable. You get much more data for your dollar, and that allows you to build expansive data sets too. And so I think that by taking a traditional assay, scaling it up using automated biology, also being smart about the assays and bringing down the cost per data point, that's what's allowing us to actually offer these differentiated services than what currently exists. And what we're seeing uptake on is customers from across the scale, actually. So large pharma is a large customer of ours. They have an insatiable need of data generation and ingesting that data into their models. They might have some of these capabilities too, but they also realize that might be locked into a silo somewhere. That might service a different group, and their group wants to do something in computational biology too that they need an unlock for. It might also be that they have overflow capacity. So some of these methods are quite in demand. And so we always have really strong relationships with pharma, been a long customer of Gingko, and we oftentimes have customers coming back. So it could be a small campaign to begin with or a large campaign, but generally over time, they want to get more and more data, and we see that now. We don't know what the biological scaling laws are for bio AI. That is, I think that the tech community, when we're talking about like ChatGPT and large language models, it was fairly well understood, or at least it was hypothesized what the scaling laws could be. That is, if you ingest more and more data, you'll get that much more predictive, right? We don't know what that looks like in bio AI. So part of this too is kind of an education for us and as well as for our clients of how much better do the models get with increasing amounts of data. And we're going to push those limits and we're going to see how far we get. Pharma has an appetite to do that, and they have the dollars to do that. Then we have other customer bases. So biotech is interested in this. A little bit harder to generate those large campaigns with them, but kind of a more focused attempt. Tech bio is kind of an offshoot of biotech that really their whole valuation is based off of how do I build these new AI models? How do I change R&D? They're much more welcoming the risk involved in that, right? And even if they have kind of these critical fundraising dollars that are tough to come by now, they want to put that into like an AI campaign because that's their whole thesis. And then you have on the other side of the spectrum, what we're seeing a lot of is tech companies, pure tech companies, large pure tech companies getting into this space as well. And so we work with customers across the spectrum, and it's always fun to see the way that their needs differ. And we could get into that a little bit, but let's just say that that we have a flexible business model that actually accommodates each one of them.
How do you keep from falling into the trap of getting commodified in AI? I mean, the AI can do more and more. More something like AlphaFold, like it's out there, kind of. It's in the open source. Like it's hard to make money off of it. Like, what are kind of some of the ways you keep people coming back, I guess, and maintain your value?
Yeah, I think you're right. There can be a commodification of models. And so we have, we've generated a number of models at Gingko. We've kind of put them out as APIs, and we put them out almost as free. Now you can get them for tokens, right? And so you can pay by a credit card. It's pretty cheap though. And there's some uptake for it, but I don't think there's much value in the models because they are so likely to be commodified. But those are general purpose models. They're not made for a particular customer, and they're not made for, personally, if I'm a pharma, for me. And so if I'm a pharma, I'm going to want to compete, and I want to be competitive in a way that my data and the designs of my data are around my strategic focus. So everything is aligned. So the data generation is going to be aligned with the modality of key choice and the targets of key interest, right? And you build that model that is suited and aligned for your strategy. I think that's where you get differentiation.
Because that's always going to be bespoke.
Exactly. And this is why pharma is often interested in differentiating from a standpoint of that data. So they want to be very prescriptive. They want the data to look exactly how they would run the assay. They would want the targets that they typically run, and they want to pay and have it be exclusive to them. So once we generate that data set, that is just going to be the partner's data set. It's all fee for service that we do. So that means that it's the data, it's really the customer's data. They're telling us the sequences, they're telling us the inputs, and we return to them the data set. We can do some additional modeling on top of that. We have partnerships like that. Or sometimes if they have an AI ML team, they'll just take the full data lake and we'll just transfer that over. And so that's one approach here. I think the other one is that, if you're in the tech bio or tech space, you may not see differentiation in terms of the way that you actually design your experiments. In fact, you see differentiation in your computational, in the architecture that you use. And you're actually more trusting the way that you're going to build the model yourself to differentiate than actually you don't need exclusivity on the data, if you will. And so they're looking for the cheapest data per dollar point possible. And they're going to take that data, and then they're going to use their own special sauce to actually do the modeling. So that's how they get their differentiation, right? Now, coming back to data points, what about us, right? To us, it doesn't really matter one way or the other. We have these flexible arrangements, right? But the key thing is that you still need the data in order to generate it, right? And in order to generate data at scale and the quality, reliability, and turnaround time that you need, we really think that we're special in that case.
Yeah. It seems like there's a lot of pressure on that data, because it's essentially training data, right, that you create. So it's got to be representative. It's got to be, I guess, kind of accurate and free of bias, and the end product has to be predictive. Like, what are some of the ways you kind of do that quality control and make sure that the data you're giving people is really, really good data?
Yeah, it's an important point because data is our currency, right? And so it has to have and currency only works if everybody agrees upon the value of it, right? And I think that in this case, everybody agrees that it needs to be high quality data. And so what we've done is for each one of our products, we've built a certificate of quality, if you will, that comes along with every delivered piece of data. Now, the way that we deliver data is on a continuous basis. So we've built these production engines that allow us to have a certain throughput every single week. And so if you have a very large campaign, we will deliver data to you on a rolling basis. Or if you have something smaller, we can spit it out to you in just a couple of days, whatever it might be. And regardless, if small or large, we're delivering it along with the QC for the data. And so the data has specific QC that we've been very thoughtful about, thinking about the bias and the types of measurements that we're taking. What is the standard deviation or the standard error certain with some types of measurements versus others? How many replicates do you need to run in this case? Can you run control replicates on the same plate, and then you can minimize the number of actual replicates of your sample so that you can get more throughput? All of these things go into the initial data design. So we kind of provide that consulting service initially to design the data with the customer to make sure that they have confidence that their input is the right thing. And then on the other side of it, after we get through the process, after we collect the data, we can deliver in a raw format. We typically have a presentation that shows it's nice and shiny too. But at the same time, it really comes down to that certificate of quality, saying that each sample, each measurement passed a pretty stringent threshold for quality.
Stepping back a little bit, there's obviously AI has been a big topic of conversation at this conference. And I'm sure many other conferences this year. What are some of the trends you see in either the technology itself and how it's evolving, or the evolution of how the industry is getting its head around the AI technology and adoption, use cases, things like that?
Yeah, it's fascinating, and it changes so quickly. The latest buzz is these MCPs, right? And these model protocols allow you to essentially hook up LLMs to other APIs, right? And so what we're seeing now is this convergence of kind of LLM sits in a box, bit of a black box to be honest, on your computer, right? And now it's starting to interact with the physical world. Even that physical world is still digital in a way, but it's interacting with CRMs, it's interacting with Slack, it's interacting with things that you actually touch every day. And so I think that's an interesting way to think about from the tech side, the way that these large language models and AI is evolving into this agent type space, right? But when you have now the space of biology, you can think about these MCPs actually allowing you to talk to our automation. And so there's a lot of discussion now internally. We have these racks, these reconfigurable automation carts. We sell them now as hardware and software. We've been using them at Gingko. We shifted models around a little bit. We realized there was an appetite for new types of automation. And so you can either buy the data service from us or you can build the thing in your house yourself. But either way, these racks are perfect for lab in the loop type infrastructure, right? And so you can imagine one day you have a large language model and an MCP that essentially talks to racks and then orders up experiments. You have an artificial scientist that comes up with hypotheses. It knows exactly what your racks can and can't do, and now it's talking and ordering data, right? That data is generated, and then it's returned back to that LLM. The LLM and the AI underneath it is ingesting this data, it's understanding this data, and now it's actually coming up with another validation, right, based on that, and a refinement of it, and then it's sending it back. So I think these lab-in-the-loop type protocols and the different tools and technologies that kind of pieces it all together, that's kind of where we're going with the field. Now, that's still several years off, but I do think that it's rapidly coming, and that's what we're going to see in the near future.
And that's really going to create a kind of a force multiplier, right?
Yeah.
I mean, when the AI is kind of like dictating another AI's experiments, everything can happen very quickly.
Absolutely, and I think that's going to be an exciting time for discovery. I think it's going to be an exciting time for getting molecules into the clinic faster, but I think it's also going to be an exciting time for hopefully improving the efficacy of these models and how of these molecules. And so the key thing here is that if you're in the biopharma space, you want to make products that end up in patients, right? I think that we're going to be really solid at the kind of the in vitro, particularly, but also this mostly preclinical work that works in these lab in the loops. I think to really close the loop, if you will, you need to extend it out into the clinic, and you need to actually show that these molecules are safe and effective as well. And I think that's going to take a slightly different turn. We're still going to need to have an integration of data, but it looks different than that initial lab in the loop. There's not going to be an MCP there, for instance, but there could be another AI agent there that is actually ingesting the clinical data if there is the right type of infrastructure to support that. And that's going to be on I think an institution by institution, company by company basis. And that's another point of differentiation. I think that as you can extend this feedback loop from your initial work that could be generated from the kind of an AI scientific hypothesis, you then spin up a molecule based off of that. And eventually there's a refinement method there before it goes into a human, but eventually it needs to go into a human. You learn from that and then it actually needs to come back, and it needs to do the full loop around for the next course of discovery.
It's absolutely fascinating. This is really, really interesting stuff. Very, some of it a little bit went over my head, but you're very good at explaining it. So thank you so much for sitting down with me, and looking forward to seeing more of what comes out of your guys' operations today.
Yeah, thanks for having me. It was a fun conversation, and enjoy the rest of the conference.
You too.
Great.